Hi, today I'm going to talk a bit about clustering and dimension reduction and just go through a few step-by-step examples of some methods and also talk about a few of the basics behind clustering. So first, why do we cluster? Well, it's mainly to show the structure and patterns within the data, especially those that are hidden and aren't generally visible at a first analysis. And then we want to classify our data into subgroups based on their similarity, such as distance. And then we aim to create these clusters based solely on the data themselves, and this is a form of unsupervised learning. So here's just a little flow diagram that represents some of the general ideas behind clustering, and that we pick which features we're going to compare, and then we find the similarity between groups of data points, and then we group those together to form clusters, and we just keep repeating this until our clusters or groupings don't change. And here's just an example of how clustering and dimension reduction was used to recreate image A from, or create image D from image A, where image C is kind of a recreation and D is adding in bits of information that were left. So there are several types of clustering techniques, and those are just partitional clustering, where the set of data is divided into non-overlapping clusters or subsets, or hierarchical clustering, where each cluster has subclusters and the dataset can be organized into a tree or nested clusters, and exclusive clustering, where each object belongs to a single cluster. Then we have fuzzy clustering, where each object belongs to each cluster, but it's given a weight between 0 and 1, as we can see here, where this green cluster and this red cluster overlap. So these points here that are only in the red cluster will just have a would have a weight of 0 in the green cluster, and the points that are in both might have a weight of 0.4 or 0.6 depending where they're located in this little region. We have complete clustering, which means every object is assigned to, as opposed to partial clustering, where not every object is assigned to a cluster. As we can see here, we have 3 distinct clusters and then points that don't really belong to a cluster. Then we have overlapping or non-exclusive clustering, which an object can belong to multiple groups simultaneously. That's just another example of fuzzy clustering. So then we have the types of clusters, and we have prototype-based or center-based clusters, so in which the objects are closer to the prototype that defines a cluster or the center of the cluster than to any other cluster. So as we can see here, that these green points here belong to this green cluster because they are closer to this green center than to any other center, regardless that they are closer to some of the red points than some of the green points. And then we also have density-based clusters, which are just regions of high density, here represented by the red, blue, and green clusters, separated by regions of low density, as we can see some scattered points here in gray. And then well-separated clusters, in which the objects are closer to all other objects in the same cluster than any objects outside the cluster. So unlike the previous slide, where we had green and red points closer to each other than to other red and green points, here we can see all the red and green points are closest to red and green points. And so I just applied some clustering techniques to this dataset made available by Howden, and that he used in this article. And here they used non-negative matrix factorization to compare expression profiles without and after antibiotic exposure, and they concentrated both on small RNAs and mRNAs. And here I just used the dataset for mRNAs, which is available on the database, and like they did in this article, I reduced the dataset, the number of genes, down to 125 by first removing any genes that had fewer than 10 reads, and then log base 2 transforming all the points and finding the standard deviation of each gene, and if the standard deviation was less than 2, then that gene was also omitted. So then which methods are generally used for gene expression data, as this is what I will be concentrating on. So there are 3 main methods, and they're principal component analysis, which is a method of dimension reduction, and it determines the dimension with the greatest variance. K-means clustering, which divides the data into clusters based on their distance similarity, and hierarchical clustering, which provides a stepwise pairing of similar data points. So first, principal component analysis, and as I said, this is shows the direction with the greatest variance, the principal component, and you can also find directions with the less than greatest variance, which may be helpful if we have a data set with many directions. So what does the PCA tell us? Well, it is a method of dimension reduction, and it can help us determine whether samples can be grouped. It helps us to identify which directions have the largest variation, but these are not necessarily the directions relevant for separating the samples, and data classes are not the same as the principal components. So where do we start? Well, the principal component is the eigenvector with the highest eigenvalue, or the direction with the greatest variance, and it allows us to choose which dimensions best estimate our original data. Here we have a data set that we'll be using in this example, and we're just going to call this matrix original. And then we're going to normalize this set, and we're going to do that by subtracting each point from its mean, and so that the new mean will be 0. And we're just going to call this data adjust. From here, we're going to find the covariance matrix of the adjusted data set, such that we find the covariance of each dimension with respect to the other. And as you can see, if we find the covariance of x with respect to x, that's the same as finding the variance. And then we just make the covariance matrix of the normalized dataset, and from our previous slides, using those x and y values, this is just the covariance matrix. So then we're going to calculate the principal components by first finding the eigenvalues and eigenvectors of the covariance matrix. So where A is the covariance matrix, we can use this matrix identity to set the determinant equal to 0 and find the eigenvalues. And then from here we can find the eigenvectors corresponding to each of the eigenvalues. So as we can see here, that the eigenvector with the greatest eigenvalue is this bottom eigenvector here, with the eigenvalue of 1.284, and this is our component. And this is the direction that best estimates the normalized data. If we make a plot here of the normalized points, and we plot the principal component here, which has the greatest eigenvalue, and then the second direction here, we can see that this one is closest to our data set. And so then we need to choose a feature vector, and the feature vector is just a matrix of eigenvectors of the dimensions that you would like, of a number of dimensions that you would like to keep. So each column in the feature vector is an eigenvector. And so if in this case we want to keep both dimensions, then we have an eigenvector that is 4 by 4, and each column here, a feature vector which is 4 by 4, and each column here is an vector, but if we choose to just keep the one dimension, the feature vector will be our principal component. So then we need to transform the data into a new data set that has the reduced number of dimensions. And we would just find the reduced data set, or the transpose of it, by multiplying the transpose of the feature vector by the transpose of our adjusted data set. And if we were keeping both dimensions, this is what it would look like, and if we were to just keep one, here's what it would look like. And so even after doing this, you can still get back your original data from these new matrices just by multiplying your feature vector and your final data set, and then adding your mean back to that later. So how do you visualize the information that you've gained from the principal components? Well, I've chosen to show a biplot, which can allow you to show how much each variable can contribute to the principal components. And here we can see, as the blue lines represent each of the different features being tested, that the length of the line is proportional to how much that factor contributes to the principal component. So here we can see this factor here greatly contributes a lot to the first principal component, as it's quite long in the x-axis, but not very much to the second principal component, as it's quite short in the y-axis. Whereas here, this factor here contributes a lot to the second principal component, but not very much to the first principal component. So, as I said, the contribution of each variable is proportional to the length and its direction. So, we can see here that even though some of these variables down here are quite long, that they don't contribute as much as their points fall below 0, and we want variables that are within this first quadrant. So next we're going to go on to k-means clustering, which is a prototype-based partitional clustering technique. So it means that it's based that we are comparing the distance to a center or centroid, and each point belongs to one cluster. So it just attempts to find a pre-specified number of clusters, k, which are represented by the centroids, hence prototype-based clustering. And the prototype or centroid is usually just the mean of a group of points, and is applied in continuous n-dimensional space. It groups the data into clusters based on distance, such as Euclidean, as we're going to use here. This is most useful when there is already a hypothesis about the number of clusters that the data should be grouped into. So the basic algorithm is that we select k random points as initial clusters, as initial centroids, and so here we just have our unclustered points, and we choose 3 random points as our initial centroids. And then we're going to associate each of the remaining points to their nearest cluster representative. Here we're going to use Euclidean distance, but you can also use other distance functions. So we can see here after the first iteration that all points have been assigned to a cluster. And as we can see that not all clusters appear as though they should. Some of these blue points might belong better to this yellow cluster. But we're going to recalculate new centroids based on these clusters, and we're going to find that the centroids have moved. And then again, we're going to reassign all of the points to clusters again by calculating the distance between them and their centroids or cluster representatives. And then we're going to keep doing this until the clusters do not change. So here, after a second iteration, we can see the clusters have changed again, and we keep repeating this process until we find that the clusters don't change. Here we can see this has been this is the best suited clustering to these points. So we're just going to go over this again as we select k random points as our initial centroids, and we associate each of the remaining points to a cluster representative, and then we're going to recalculate the new centroids and keep repeating steps 2 and 3 here until the clusters do not change. So it looks a bit like this. So how do we know that the clusters are right? Well, we know that optimal clustering occurs when there is a global minimum sum of squares error, and suboptimal clustering occurs when we reach a local minimum. So this is often due to the initial centroids being randomly chosen and can be avoided by running multiple replicates, each with a different initial centroid. We can also update our centroids incrementally rather than after all the points have been assigned, which reduces the risk of having an empty cluster, but introduces order dependence. So, determination of clusters. So if we run multiple replicates, the clusters that are right are determined from the replicate with the smallest distance between centroids and points, or sum of distances. So as we can see here, we ran 5 replicates, and each one, depending on where the random centroids were chosen, had a different number of iterations and a different total sum of square distances. And as we can see here, that the best replicate was replicate 3, which had the smallest sum of square distances. And this doesn't necessarily mean that it had the smallest or largest number of iterations either. As you can see here, it had 12, which is in the middle. And so this is just the clustering that we got from replicate 3. And we can see here that we have 6 clusters. They're not distinct as some of them run into others. And since some of these clusters run into others, we want to know how valid these clusters are. So we can validate these using a silhouette map. This just assigns every point in its cluster a value between -1 and 1, depending on how well suited that point is to that cluster. The value of 1 means that that point absolutely should belong in that cluster, and a value of -1 means that that point should belong in a different cluster. So if we see our silhouette map here, we can see that the point in cluster 5 has a very high silhouette number, as well as a lot of points in cluster 4. And if we just look at that clustering again, we can see here that these are a lot of the points in cluster 4. Cluster 5 is here on its own, so it's not very likely for that point to belong to another cluster. Whereas points here in cluster 3, which have a quite negative silhouette value, and are here in the red, they're in the middle of cluster 4, and maybe we should try a different number of clusters, or try maybe a different clustering method. So what do we know is in each cluster? Well, to find out which genes have been assigned to each cluster, we can use a heat map to see which genes are in which cluster. So where 0 is that gene isn't in that cluster, and 1 is that the gene is in the cluster, each of these small lines represents a gene. The gene names are not presented, as it can become a bit, as the heat map can become busy, but we can see here that we have that one gene, that one point in cluster 5 belongs to this single gene, and all of these points in cluster 4 are all of these red genes here. And so then we'll move on to hierarchical clustering. And this creates multiple clusters where the data is ordered into a hierarchy of similarity, and they can be displayed as a dendrogram or tree, or as nested clusters to show the relationship. Here is the nested clusters are just grouping those similar groups together, and here the dendrogram or tree, which just shows the nodes where different points can branch off. So how do we determine this hierarchy? Well, there are two methods. The one is agglomerative clustering, and the second is divisive clustering. In agglomerative clustering, we start with each point belonging to a single cluster, and we repeatedly merge the two closest clusters until all points are encompassed in one large cluster. With divisive clustering, we start with one large cluster and we split the two most dissimilar clusters until each point is in a separate cluster or a specified maximum number of clusters has been reached. So how do we determine cluster proximity for hierarchical clustering? Well, we can calculate the proximity in 4 ways. Single linkage, which is just the distance between the two closest points in a cluster. The closest points between all clusters are calculated, and then those that are closest, the clusters with the shortest distance, are grouped. And then we have complete linkage, in which the furthest point between two clusters is calculated, and again, the clusters with the shortest distance between them are grouped. And then we have average linkage, in which the distance between all points in all clusters is calculated, and the two clusters with the shortest average distance are then grouped. So the distance between all clusters and all points and all other points are calculated, and the average between each of the clusters is determined. And lastly, we have Ward's method, which is a centroid-based method in which the mean or the centroid of each cluster is found, and then the distance between means in different clusters is calculated, and clusters with the closest centroid are then grouped. So, as we can see, we have these 4 methods of calculating the distance or proximity between clusters, and when we visualize these 4 methods, we can see that we get quite different results in the way that they cluster. At a glance, we can see that average linkage, complete linkage, and Ward's method have dendrograms of similar shape. But if we look closer, we can see that here in average linkage, points 19 and 3 have been paired together and determined as not being able to be clustered with any of the other points until the last clustering opportunity, as they are in complete linkage, or it's the same. However, in Ward's method, point 19 has also been determined as one of the last clustering options, but however, point 3 is far down here and has been clustered quite early. Some other methods of clustering or dimension reduction that are often also used with gene expression data are fuzzy C-means clustering, where points often belong to multiple clusters, and non-negative matrix factorization, or NNMF, in which an original matrix is factorized into 2 separate matrices which estimate well the original matrix but lose some of the original data. So fuzzy C-means is similar to K-means clustering, but is non-exclusive, such that each point is assigned a weight between 0 and 1 to each cluster, and the data points can belong to multiple clusters. As we can see here, that we've applied fuzzy C-means clustering to the same dataset, we get a similar pattern, where each of the colors represented is actually the cluster that has the highest weighting for that data point. So as we can see here, we have a kind of similar pattern to the K-means clustering, where we have lots of a few outlier points and several points here in the middle that have been assigned to one cluster but could belong to the other or another. So when we compare k-means and fuzzy c-means by, say, comparing their heat maps, we can see that where genes solely belong to one cluster in k-means, here that in fuzzy c-means they can belong to multiple clusters, which is helpful for when we're clustering gene expression data. So lastly, we have non-negative matrix factorization, and this just splits the original data matrix into two factor clusters such that when they're multiplied together they get back a matrix of the same dimensions. And W and H, our factor matrices, are dependent on a pre-chosen number of classes, K. And so just to recall that if X is an m by n matrix, then when we factorize, W is an m by k matrix where k is the classes, and H is a k by n matrix, so that when we multiply them together we get back an m by n matrix. And each of the columns of W define a metagene in this example, or a pattern of gene expression, and each of the columns of H represent the metagene or pattern of gene expression for a corresponding. So given that X is 125 by 20, our number of genes and our number of samples, we can find and the K classes that we've been using, that can find a heat map of a log2 base transformation of W, as some of these values are quite large, which is then going to be 125, so each of our genes, by 6 our classes, or the different patterns we're looking for. And then a heat map of H finds that here is our 6 patterns we're looking for, and the 20 samples, and how they are expressed in each of these patterns. So what does this tell us? So for matrix H, the samples can be grouped in K clusters, and the clusters of each sample are placed into corresponding clusters depending on which metagene or pattern is most highly expressed in the sample. So here we can see that we have a peak here for cluster 4, this blue cluster, at sample 7. So if we go over here to sample 7, and we go up to cluster 4, we see here this got a very high heat map value. And so this can just tell us which samples expressed more heavily in certain patterns. So here we can see here cluster 5 has a peak here at cluster 12 and another one at 14, and we can see at 5 that we have peaks again, so maybe those samples induce similar responses. So this can also be done to the genes, not just the samples. So by doing this with a W matrix, we can determine a cluster for each gene. But as we can see that very quickly, that these plots and heat maps get quite busy, and they can be quite hard to determine. It can be quite hard to determine what these plots are telling us. If you see here that in cluster 1 we have a peak here over near cluster maybe 30 and a few smaller ones just before that. And so if we find around cluster 1, we have here a quite dark peak here. That's around about 30. You can see that maybe gene 30 is definitely expressed in pattern 1. But maybe limiting this type of visualization to maybe 20 samples of 20 genes might be a good idea. As we can see, it's quite busy. So here I'm just going to leave with a few links for further reading for the k-means and hierarchical clustering, fuzzy c-means, and non-negative matrix factorization. Thank you.